Hi, my name is Lucy, and I was interested in neuroscience and genetics. So when I decided that I wanted to conduct some research, I wanted to be able to combine those two. And so for my project, I researched how altering EAT1 expression to manipulate glutamate uptake affects aggression in Drosophila melanogaster, or the fruit fly. So first, why should we research aggression? Well, ecologically, researching aggression can provide us insight into the interactions within species. This includes aggressive encounters between large animals, but also small animals too. However, what is probably more relevant to all of you is aggression's presence in our society. So aggression manifests itself at elevated levels in several mental illnesses, including depression and bipolar disorder. And so by researching aggression, we can hopefully better understand the neurological basis behind these mental illnesses and potentially develop better treatments to treat these mental illnesses. So my project researches aggression in Drosophila melanogaster, or the fruit fly, because aggressive behaviors in the fruit fly are well defined and easily identifiable, and most importantly, the fruit fly shares between 60 to 75% genetic similarity with humans. This means that aggression research in the fruit fly could potentially be applied to the genetic regulation of aggression in humans. So specifically, my project researches glutamate's influence on aggression. And glutamate is a neurotransmitter, which is a chemical used by neurons or brain cells to signal to other neurons. And so this is exemplified in this diagram up here. So the top neuron has just released glutamate, which is the purple dots, which will then be received by the bottom neuron with the message to carry out a specific function in the brain. This is a continuation of research that I conducted last year, which found that glutamate is expressed in some of the neurons of another neurotransmitter called octopamine. And this diagram down there is what that looks like in the brain. So this neuron has the potential to release both octopamine and glutamate. The primary finding of my research last year was that when glutamate was decreased, specifically in these octopamine neurons, aggression decreased. This year, my project built upon this research by manipulating glutamate expression in a different way. So in other words, rather than manipulating glutamate expression in octopamine neurons like I did last year, I did so in a different set of cells. So as a continuation project, my guiding question is, how does glutamate contribute to aggression circuitry? And my hypothesis is that glutamate promotes aggression. Although my results from last year did confirm this hypothesis, they only applied to glutamate expression in octopamine neurons, so this hypothesis is still relevant to a greater expression of glutamate. My approach to answering this question was to increase and decrease glutamate expression by genetic manipulation. And I predicted that if my hypothesis was correct, then when glutamate was decreased, aggression should decrease, and when glutamate was increased, aggression should increase. So I manipulated glutamate expression by altering expression of one of its transporters called EAT1, which stands for excitatory amino acid transporter 1, but you don't need to understand what any of those words mean to understand this research. You just need to know EAT1's function in the brain. So as I already mentioned, EAT1 is a glutamate transporter in the fruit fly, which means that it regulates glutamate signaling in the brain by moving it to different parts of the brain. Specifically, it is primarily expressed on glial cells, which provide support and protection for neurons. And from its position on these glial cells, it recycles glutamate present in the gap between two neurons to return it to the neuron that it was sent from, in this way regulating glutamate's expression or signaling in the brain. So by using EAT1 to manipulate glutamate expression, our knowledge of glutamate's influence on aggression can be expanded and differentiated because EAT1's expression in the brain is different than that of glutamate in octopamine neurons, which is the expression I manipulated last year. So to visualize this different expression, I dissected a fly brain expressing EAT1. And so on the right is an image of that dissection, and on the left is an image of the fly brain and a fly's head to provide context. In this dissection image, each red dot represents the nucleus of a cell expressing EAT1. So if you look at how many red dots there are in this image, you can see that EAT1 expression in the brain is pretty widespread. Comparing this to glutamate expression in octopamine neurons, which again is the expression I manipulated last year and is shown in these two small green regions here, you can see that EAT1 expression in the brain is much more widespread than glutamate expression in octopamine neurons. So this means that the amount of glutamate expression that I'm manipulating this year compared to last year has expanded, and as a result we can look at how this expanded manipulation of glutamate expression affects aggression, in this way expanding our knowledge of glutamate's influence on aggression. Using EAT1 is also significant in our approach to increasing expression of glutamate, otherwise known as overexpression, in that previous approaches to overexpression of any neurotransmitter have done so intracellularly. The problem with this is that although an increased expression of the neurotransmitter may increase inside the cell, this does not necessarily mean that an increased expression of the neurotransmitter will be released. But by using EAT1, which is expressed outside the cell on glial cells, we can skip the question of release and instead focus on glutamate uptake, or glutamate being received by the responding neuron. We can do this by decreasing expression of EAT1, so less glutamate than usual is recycled and more is available to be signaled throughout the brain. Using this method, we can also reduce glutamate expression by overexpressing EAT1, so more glutamate than usual is recycled and less is available to be signaled throughout the brain. So in conducting these behavioral experiments, I isolated male flies at the pupal stage so they could gain no prior social experience. Then 3 to 5 days after they had matured into adults, I introduced 2 male flies of the same genotype to an aggression chamber, which is essentially just a plastic box with a food cup in the middle of it. And I filmed their interactions for 90 minutes. I then scored those interactions for the following parameters, the first of which is the latency to lunge, or how long it takes these flies to start fighting. The second is the number of lunges, and a lunge is when a fly rises up on his hind legs and snaps down on top of the opposite fly. Third was the number of wing threats, which is when a fly raises his wings up and out to make himself look bigger and more threatening. And finally, number of wing extensions, which is when a fly raises his wing at a 90-degree angle towards the opposite fly. So my first approach to answering my question was to increase EAT1 expression or decrease glutamate expression, and in these experimental flies, which are shown in purple, we see an increase in latency to lunge compared to the controls, which received no genetic manipulation. So this means it took these flies longer to start fighting. We also see a decrease in number of lunges, a decrease in number of wing threats, and a decrease in number of wing extensions. So overall, this is a decrease in aggression, which suggests that EAT1 overexpression or reduced glutamate expression decreases aggression. This supports my hypothesis by suggesting that at regular levels of expression, glutamate promotes aggression. Additionally, in these results, aggression decreased to the same extent as when glutamate was decreased only in octopamine neurons, which indicates that expression in octopamine neurons is glutamate's primary contribution to aggression. In other words, in this experiment, glutamate manipulation in cells or neurons other than those of octopamine ultimately didn't have much of an effect on the aggression phenotype. So finally, my last approach to answering my question was to decrease EAT1 expression or increase glutamate expression, and in these experimental flies, which are shown in pink, we also see an increase in latency to lunge and a decrease in number of lunges. And we see what looks to be a decrease in number of wing threats, though this isn't yet statistically significant, and no change in number of wing extensions. So overall, this indicates that reduced EAT1 expression or increased glutamate expression decreases some aggression parameters, which does not support my hypothesis by suggesting that at regular levels of expression, glutamate inhibits aggression. However, EAT1 is expressed at the neuromuscular junction, which is the point where a motor neuron signals to a muscle cell. So this means that the aggression phenotype that we see in these results could reflect an impaired muscle capability. So at the neuromuscular junction, as EAT1 expression decreases, the resulting increased expression of glutamate could be telling the muscle cell to contract for a prolonged period of time. This means that, or this could tire the muscle cell out and reduce the fly's ability to lunge. So to address this, I will be excluding EAT1 manipulation below the neck, which is where these motor neurons are located, and what would be the spinal cord of the fly. It's actually called the ventral nerve cord. To determine the effect that EAT1 is having at the neuromuscular junction, as well as to determine accurately glutamate's influence on behavior alone. I will also be limiting EAT1 expression to octopamine neurons to determine if the small percentage of EAT1 that is expressed on octopamine neurons, or that is expressed on neurons, is expressed on octopamine neurons, and whether that expression is important for the regulation of glutamate octopamine signaling in aggression. So because the fruit fly is genetically similar to humans, this research could potentially apply to aggression in humans. Specifically, it could be that abnormal glutamate circuitry is responsible for increases in aggression due to injury or disease. And if we can confirm this, then this research could help us potentially develop better and new treatments and therapies to treat aggression under these elevated conditions. So I'd like to thank my mentor, Dr. Sarah Certel at the University of Montana, Lizzie Catudio-Garrett, Ned Brewer, and other members of the Certel lab at the University of Montana, and my APS teacher, Rob Jensen, at Hellgate High School. Here are my references. Thank you.